Welcome to Lecture 13.1. We're going to provide an overview of gene regulation. Gene regulation refers to the ability of cells to control their level of gene expression. I think it's important to establish this relationship, define these terms. A genome, we know, represents the entirety of the genetic material within the cell. A proteome represents the entirety of the proteins expressed in that particular cell. All cells of the human body have the same genome, but yet not all cells of the human body have the same proteome. A cell has to have the ability to express proteins at certain times and in specific amounts in order to establish itself, in order to adapt to its environment, in order to succeed. And so I think this is going to be very important when we start to talk about how a cell, how a living thing is able to adjust and adapt so quickly, how can it be so flexible? It's really at the level of gene expression where most of that occurs. Now I want to define what a constitutive gene is. A constitutive gene is a gene that's not regulated. And the reason it's not regulated is because we need it at constant levels. So think about these genes as producing products. And when we talk about products, we're talking about RNA or protein that are needed throughout the cell's day, throughout the cell's lifetime. So I'm thinking like ribosomal RNAs, for example. We need ribosomes constantly in a cell. So they're going to have some baseline level of expression. Now, they can be upregulated in bacteria in response to new metabolic conditions. But for the most part, they're going to be expressed at some low levels constantly. Polymerases would be another example. So gene products that are needed throughout the day, if you will, are going to be constitutive. Those that are only required under specific conditions. So for example, lactose is a sugar that can be metabolized. Lactose is not present all the time. So we're not going to make lactose enzymes unless lactose is present. That would be a regulated gene, and it's going to be the focus of this chapter. So what are the benefits? First is the conservation of resources. Listen, we just got done with chapter 12 talking about how energetically expensive the process of translation is. We burn through ATP when adding amino acids to tRNA. We burn through GTP when establishing the initiator complex. We burn through 2 GTPs during the elongation process. I mean, this is energetically expensive. So why produce a protein unless it's absolutely necessary at that given time? And it's not only energy, but it's also resources, raw materials. I mean, there's only so many amino acids a cell has. Why have those amino acids be stuck in a protein that's not needed at that point? Why not make those amino acids available for those proteins that are required in order to adjust to its environment? The other thing that we think about is development. Development of a cell, the development of a multicellular organism. I mean, this is a highly regulated process because if you recall, at some point you were a single-celled zygote who was diploid due to the donation of a complete set of chromosomes from your dad and your mom. And so the fertilization process occurred. But that zygote ultimately became the cells of your heart, of your skin, of your nervous system, of your liver. I mean, go right down the line, all the various cell types that you see. How can that cell, that basic progenitor cell called the zygote, become all of those different cell types? The genome doesn't change. The genome is established during the fertilization step. You are diploid as soon as that sperm and egg successfully fuse together. So how can we make all these different cells when the information doesn't change? The answer is how the information is expressed and the times they are expressed, the levels of expression, all help then establish a multicellular organism with very diverse cell types. Now, for prokaryotes, that's not necessarily an issue because it's single-celled. And so all of the basic functions of that prokaryote occur within that single compartment. Now, one of the things that you learned about in anatomy and physiology, or you may have heard about, is this idea of homeostasis, right? Balance. And as a multicellular organism, I have organ systems, like the digestive system, respiratory system, excretory system, nervous system. All of those things contribute to trying to help balance and maintain steady state levels of waste products, of oxygen, of nutrients, body temperature, we know is important, etc. Bacteria are not that fortunate, right? Because they don't have all of these different systems working together. They're an entity as a cell, they're an entity unto themselves. Bacteria are always faced with changing environments. There's not much stability when you're a bacterium. And so they're constantly responding to changes by regulating gene expression. Now you may say, well, I'm not interested in bacteria, I'm interested in the human experience. But there are a lot of correlations we can draw between how a prokaryote expresses their genes and regulates their genes with eukaryotic examples. And so I think it's important to understand how it happens in bacteria and prokaryotes in order to draw then and establish relationships, in order to draw conclusions with eukaryotes. One of the examples we're going to see is how bacteria can adjust when lactose is present. When lactose is not present, E. coli doesn't make proteins involved in the metabolism of that particular sugar. It doesn't make proteins involved in its transport. But when lactose is available, now the bacteria is able to upregulate gene expression in order to import lactose and in order to digest lactose in order to make energy. Here's a simplified example. We're going to talk about this more. But we can see here that at its resting point, what I mean by that is when no lactose is available, we can see that there's very little in the way of lactose protein being on the surface and very few lactose enzymes available in a cell. However, when lactose does become available outside extracellularly, that lactose then is imported and acts as a signal to allow the cell to change its physiology through gene regulation to produce many more copies of lactose permease and many more copies of beta-galactosidase. With the help of the transporter, much more of that lactose is now available in the cytoplasm for degradation or metabolism by that enzyme. When lactose is used up, either degraded or stored away in another form, we can see there's no longer a need to have so many copies of each protein. So now the bacteria is going to degrade those proteins, recycle those amino acids, and now wait for lactose to become available again. So this makes a lot of sense. Now you notice it's not all or nothing, meaning there's no copies of those proteins available, and then all of a sudden there's a million copies. We can see there are very few protein molecules available, enough to bring a few molecules of lactose in to serve as the signal to now allow gene expression to be regulated. Now in eukaryotes, very different because it's not so much about metabolism. Bacteria live to eat in order to get enough energy to reproduce. In eukaryotes, when we start to talk about gene regulation, we're really talking about cell differentiation as the major point of control. Because as we've established, the same genome in those many cell types does not ultimately produce those different proteomes. Because there are different identity of proteins made and differing amounts of that same protein. And so that's what allows a heart cell to function so differently than a nerve cell. It's not the genome, it's the proteome. And so here's a great example. We know skeletal muscle cell is involved in contracting, has to be able to shorten and then lengthen in order to support activities. Well, that's very different than a nerve cell, which just at its structure, right? You just look at its form is very different. And notice these long extensions that are able to receive and send signals. Very different than what a muscle cell would do. And then here's a skin cell, different from the other two. Skin cell aids in protection, has a certain shape, a certain arrangement of how it's packed together with other skin cells in order to act as a barrier. What allows all of these to be so different is not the genome, but its differences in gene expression, which ultimately is going to help establish the proteome. And so developmental gene regulation in mammals is observed during the development of a fetus. So we're constantly refining body parts. Body parts are constantly maturing. We're increasing in size. How does that happen? Well, gene regulation. A great example is seen in hemoglobin, which we know plays a role in transporting oxygen throughout an organism. Fetal hemoglobin is different than adult hemoglobin. It has a higher affinity for oxygen, which allows for a more efficient transport of oxygen from the maternal blood to the fetus. And so during the earliest stages, embryonic stages, the hemoglobin protein has much different components than the fetal hemoglobin and the adult hemoglobin. During those early stages where, man, I need lots of energy in order to have my cells differentiate, I have the highest affinity for oxygen in order to help support respiration. As it matures through the stages of the embryonic stages to the fetal stages, the differences in makeup of the hemoglobin changes its affinity. It still has a high affinity for oxygen, but not quite as high as the embryonic stages. Now let's compare that to the adult hemoglobin, which is made up of 2 alpha subunits, 2 beta subunits. We have moderate affinity for oxygen because the requirement is not as important as it is during the earliest stages of your life. And so how does this happen? Well, simply we turn on the genes at the different stages of life that are required to build that hemoglobin. And so we can see it's very important as we transition from embryo to fetus that certain genes get turned off and certain genes get turned on. So this is very well coordinated, and it's very important in this example of cell differentiation and development. So where does gene regulation occur? Well, we spend a lot of time here on transcription, and we're certainly going to do that in this chapter. So how a gene gets read into mRNA is one of the ways which we can ultimately impact the amount of protein. But we're going to see there's other levels of regulation as well. We can control the ability, for example, of a ribosome to efficiently translate mRNA. That's certainly a level of control. We can modify a protein once it's made. We can add things. We can add sugars. We can add phosphate groups. We can regulate its activity post-translationally. And we're going to observe these different levels of gene regulation in bacteria. Notice what we're missing here is a lack of a nucleus, right? Because this is bacteria. We're also missing the level of RNA processing, which we see in eukaryotes. So that additional level here of RNA processing makes eukaryotic gene regulation a bit different. Eukaryotes can regulate the ability for mRNA to be transported from the nucleus. It also is going to add things and take things away from that primary transcript, which ultimately is going to impact how that protein functions.